Hello everyone, welcome back to my channel. Actually, I am solving CSIR NET Life Science questions. I thought that I should discuss this question with you also. This is a question, it has come recently, 2024 March first shift, and it's a match the following, and this question is from molecular biology, a very common topic, DNA replication. And basically we have to match the functions of the enzymes that function is also similar with eukaryotic DNA replication and prokaryotic DNA replication. So here is the E. coli genes, which is prokaryote, and there is eukaryote. So there is DNA B, DNA C, beta clamp, DNA G. We have to match this X column with the Y column. Okay, and we have to identify the correct function of DNA B, like DNA B is act similarly with polymerase alpha, polymerase alpha or CDC6 or PCNA or MCM complex. But you have to know the function of each and every enzyme firstly. So for your help, I prepare a chart. Let's see the chart. So here is the chart in which I have incorporated two process, like prokaryotic DNA replication as well as prokaryotic DNA replication and eukaryotic DNA replication. And see, here is the similar functions of the enzymes, like DNA A for prokaryotes perform the similar function with ORC in eukaryotic DNA replication. This DNA A and ORC, their action is same, like they are responsible for the initiation of the DNA replication. So DNA A binds to origin C and initiates DNA replication by unwinding the origin region, and ORC also binds to the replication origin and to initiate DNA replication. Okay. Next is the helicase. Helicase is also known as DNA B. It generally unwinds DNA. Okay, and unwinds DNA at the replication fork. And here is the helicase, that is the MCM. MCM unwinds DNA double helix during replication. Okay, so you can see that within that chart we have DNA B and MCM also. So we have to match. We know the function of DNA B and MCM is same. They both unwinds DNA strands at the replication fork. Okay. Next is the primase, that is DNA G. It synthesizes short RNA primers, like and here is also primase, one primase like DNA polymerase alpha, which also synthesizes short RNA primers. So for in eukaryotes, it's DNA polymerase alpha that has same action with the prokaryotes, that is DNA G, that is primase. So primase and DNA polymerase alpha has the similar action. They both synthesizes short RNA primers. Okay, and let's see the question again. See what we get here. Firstly, here is DNA B. We have seen that DNA B and MCM complex has the similar action. Okay, so DNA B and MCM complex. So we have to select A4 here. A4, like see, option 2 here is A4, option 3 here is A4. Now we don't know which one will be the correct answer, option 2 or option 3. To know this correct answer, to choose the correct answer, we have to know the all functions of these given enzymes, right? So DNA C, I know that its function, it is similar with eukaryotes CDC6. So DNA C it's act similarly with CDC6. Okay, so B2. So here select D, A4, A4, B2, okay, and C, that is the beta clamp, it is pCNA, because beta clamp, it has this sliding clamp activity, okay, it match the DNA with the enzymes, okay, and pCNA also perform the same function. So beta clamp and pCNA, okay, and so that means C3, and lastly the remaining one, DNA G, that is D, it is act as a polymerase alpha primase activity, so D1. So the correct answer is 3. Let's check that we have selected this correct answer or not from that chart. See here, firstly DNA G, that is primase, and it synthesizes short RNA primers, and it's act similarly with DNA polymerase alpha, which also synthesizes short RNA primers for eukaryotic replication. So D1 is correct, okay. And there is also DNA B, which has the helicase activity, it unwinds DNA, and MCM, it is also having helicase activity and it is also unwinds DNA. So DNA B acts similarly with MCM, okay, we get it. And we now have to see that, yes, beta clamp holds DNA polymerase 3 firmly onto DNA, okay. So sliding clamp activity, and it is also similar with pCNA for eukaryotic DNA replication. pCNA holds DNA polymerase delta and epsilon onto DNA, because this delta and epsilon, these they synthesizes the lagging strand and leading strand, right? So increases processivity and coordinates replication and repair polymerase, okay. So beta clamp is coordinates with pCNA, and see here DNA C, it is also coordinates with CDC6. DNA C is known as loader protein that helps helicase or DNA B bind to DNA during replication initiation. And CDC6, it is also involved in loading MCM helicase onto DNA at the origin during replication licensing. Okay, so we get this beta clamp, DNA C, okay, match it, and here also DNA B helicase and DNA G, okay. So ABCD is confirmed. So from here DNA B is responsible for MCM complex, because they both have the helicase activity. DNA C, they are the loaders, so that is why DNA C and CDC6 are similar in fashion, okay. So DNA B and MCM, DNA C and CDC6, beta clamp, they are sliding clamp, and beta clamp and pCNA, okay. And DNA G, which act as polymerase alpha. DNA G act as a primase, and it also act similarly with polymerase alpha, okay. So thank you.